All right, welcome to week one. Hopefully by now you've watched the welcome video and viewed the email that I sent out with all of the links that you need. So here I am at the course GitHub, and I kind of went over, I touched on that there's the website and then there's the textbook. I think that I forgot to touch on the syllabus. There's not really a whole lot to it. I think that I explained everything already in the welcome video, but there is a syllabus. It just kind of goes over the objective, the outcomes, and the structure. Basically, it's a tutorial-based course. Complete the assignments, and that's it. The coding assignments make up 100% of the grade, and there is the final project for the advanced R2 credit section, and that represents 20% of the portion. So, with that being said, there are no exams, there are no quizzes. It's just completely based on the coding assignments. So, week one is plotting customization tutorial. So this is really just based in base plotting and just customizing them to an extent that we hadn't done previously. So I'm going to go ahead and open up the textbook here. And I already have R opened up in a separate window. And I'm going to go over here, go to the textbook, and I'm going to go down to plot customization. And the other way, I didn't have to go about it that way. I could just go down to week one, click plot customization, and here we are. Same place. So the entire purpose of this is just to show off that there's a lot that you can do with just base R plotting. We're going to touch on some different packages and visualization techniques later on, but we really need a solid foundation in base plotting and base customization before we do that. So we're going to make this plot today. And the way that we're going to do that is, the first thing that we're going to do is we're just going to refresh some of the base plotting, and then we're going to expand on it and really get into the customization. So the whole purpose here of this quick exercise is just to go over the creation of this plot. So I copied some stuff over, and now what I want to do, copy that over, and just going to run that. Cool, not a whole lot there. Copy over what we need. Cool, okay, there's the plot right there. Now we're going to add in the extra arguments to make the plot function some extra arguments to the plot function to make it prettier. So right here, so all we did here is this is our plotting function, just a very simple plot. We have an X and a Y. And we're just going to add a ton of arguments here. So let's go over some of the customization. Boom, there we go. Now this looks a lot better. We added a ton of customization, some coloring, some different points, and we added some types. So let's go through this one by one. So plot takes 2 arguments off the bat. And remember, you can use help. If I'm down here in the console, you can run help on plot, or you can do question mark plot, doesn't matter. And it just takes an X and a Y argument first and foremost, and then all these extra arguments right here. So these are just extra graphical parameters. That's everything you see here, the PCH, the col for color, the BG for background color, the CEX, the XLab, the YLab, so on and so forth. And I'm just zooming in here so it's a little bit easier to see. So we define our X as big X right here, our Y as big Y right here. The type is B for both. How do I know that? You scroll down a little bit. P for points, L for lines, B for both points and lines. So that's why in the plot you have both these points and the lines. PCH. This is plotting character or point character. So if we come over here, there is no PCH going on right there. Let's see if we can bring it up. Add points to a plot, PCH. So now, PCH is the plotting character. It's the symbol to use. And these are the plotting characters right here. So 0 would be a square, 1 would be a circle, so on and so forth. When you get down here, 15 is a square, but it's a filled square. 16 is a circle, but it's a filled circle. And you can actually go into 21, 22, which 21 is a filled circle that has a different color for the edges and the fill color. And then 24, which is what we use, which is a triangle that has an edge color and a fill color. Our edge color is blue. Our fill color is yellow. And so yellow is just the background color, so that's why we have yellow here, and then we have blue elsewhere. Notice that blue is also going to be the line color here because we used both, the B for both points and lines. So it's covering the blue color stands for the blue for both the lines and the outer edge of the points. So come down here, I kind of break these down a little bit further. CEX is the size of the points. So 1.6 adjusts the ratio or how large these points are relative to normal. Normal is just 1. So we are at 1.6 times the normal. We have X label, which is just X, Y label, which is Y. We can color the labels to make it orange. We enhance our line width from 1 to 1.4. The line type is 2. 2 means dashed. And so these are all different, just extra arguments that really enable you to customize a particular plot. So there's a lot of different things that you can do here. So we have the line width, the line type, and the line type you can do solid, you can do dash, you can do larger dashes, and so there's different resources online that you can go to find more out about that. The other way is just to try it out. So if we wanted to do this with line type 1, now we have a straight line. If we want to do it with line type 3, be a dashed line, but it's smaller dashes. So you can play around with it, but this is really the customization right here. And then main is our title, X versus Y. So now what we want to do here is we're going to separate these two plots. We notice how above we used both. Now we want to separate this out and make it a little bit more customized. We want to add a line plot first and then add a points plot over top of this. Why would we do this? Notice how earlier I talked about the line color and the edge color. So we have edge color of blue, line color of blue. What if we wanted those to be different? Here's how. We're going to first plot a line plot, and then we're going to add points to that instance. So here's our line. Okay? Now, we're going to add points to the plot instance. And this function will automatically do that. So now we have the points, the point values right here, and notice how here's my color. I can change this now. If I wanted to make it, let's make it red. Now we have red edges with a yellow fill and a blue line. Then we can also add an ab line, and this is just a constant line that we can add in for any value. So we're going to add it. So we have V for vertical. So if we run a help on ab line, H is horizontal line, V is vertical line, and it's just the values that they're going to be at. So we want a V of 10, we want a vertical line at 10. Boom, just a vertical line right there at 10. So this is really useful if you're running a mean, if you want to add a mean to a particular plot, or if you just want to add a threshold of some kind. It's pretty easy to do. And then the last thing that we can do is we can add a legend. And this is actually, copy and plot this over. So we added in this legend here. We said, okay, I want it to be top right. And yes, legend actually takes these arguments. It has top right, top left, bottom, center, and you can run a help on legend to find that out a little bit more. It takes a ton of arguments, and you can also provide specific coordinates if you don't just want to use a relative position. So sometimes that's useful if this were to be covering up any particular points, or if there was a large scatter plot that had the corners covered, the center covered, but there was like maybe a little window somewhere random, you could actually specify specific points. And the way that you would do that is like this. So now we added the legend at x equals 15, y equals 3.6. So that's how you can do that. You can actually specify that, but sometimes it's just easier just to say, you can just say, see if it'll take center. Yep, center. And so it'll actually just take that right there. But the legend has a legend argument, which is the name. And notice how we have to concatenate this. And this is useful if you're ever trying to plot two different things. Oftentimes we're trying to plot multiple things in the same image. So you might have multiple things that need a legend for. So if we wanted to legend an X and a Y and a Z, and then we have the PCH that we want the legend to represent. It's the same PCH, the point character we wanted up there, right here. The color is blue, point background is yellow. Again, we can change this to represent our updated red. And there we go. So I'm still in center here, but it doesn't matter. You can do that any form of that. So we can see that pretty quickly we can customize a ton with just base R plotting. No special packages. This is just part of the base package that is automatically loaded whenever you use R. So now I want to add a little bit of a more, there's nothing really to this plot. There's no purpose of it other than to show off different things that you can do. So this is showing off all of the different line types that I discussed earlier. It's showing off all of the different point types that we looked at in the help function. And so now instead of copying this over where I highlight it and hit Control-C, I'm just going to click this button, which will just copy it over for me. And now when I plot this, this is actually just showing off each of the different PCH values, each of the different line types. So this is just kind of a helpful little image to show off just different colors and the like. And notice how we have to use the colon. So we want to plot 1 to 25, 1 to 25, and we want the PCH to be 1 to 25, the colors to be 1 to 25. So that is how we're doing that here. And you can notice that we can add a grid using the grid function, and we added a background color to the legends. And yeah, there's just a ton of customization you can actually use here. And it's just, it's just finding out what you want to do, what you want to make with the plot, and what represents a particular plot best. So that was just with the base plot, which is useful for line plots or scatter plots, or just something that is best on a scattered grid with an X and Y bounded variable. However, what about bar plots, box plots, pie charts? All of these can just very similarly to this one, they can also just be loaded straight into R. They're already loaded straight into R, and they can be used without loading any other package. So now what we're going to do is we are going to make a fake data set of those who are infected by coronavirus. So we're going to do that using the sample function, and we're going to turn these into factors so that they have labels to them. So let's go ahead and we're going to create an infected status object. Print that out. So we just created a random sample right here. We wanted it a size of 50, so we wanted 50 long. And then we wanted to make it into a table. So we made it into a table based on the different level that the factor function created. So we have 12 infected, 23 recovered, and 15 susceptible. So let's actually go ahead and let's create another random sample of genotype. So let's say that their genes are determining whether they're susceptible, infected, or recovered. Or let's just put some extra data here. So now we have our R, capital R, lowercase r, and capital R. We can combine these two into one table, like so. So now we have now split our infected, recovered, and susceptible into these different values right here. And so if you added all these up, we'd have the same number of infected up here, which is 12. So 4 plus 6 plus 2 is 12. And the same number of RR, which is 14. So 4 plus 6 plus 4 is 14. So it kind of split those up for us. And this is okay because we don't necessarily care about this data set. It's just for this plotting purpose. So now what we're going to do is we're going to create this test here. And we are going to, what we're going to do is we're going to run this without the factor. So just a sample. This is what it looks like right here. So this is just showing that we really want this to be a factor. Why? Because otherwise it's just considered a character and we can't count it. So now we're just going to make test 2, which actually uses a factor function here. So now we have these levels down here, which enables us to count all of the specific instances of infected, recovered, or susceptible. So notice that the difference in class here, you run a class on test, you run a class on test 2, there's differences, it's character versus a factor. So with this particular information, we can create plots such as a bar plot, a box plot, or a pie chart. And what we're going to make is we're going to make this image which shows the distribution or the different makeups of each particular group. So, and we're going to combine all of these plots into one image or one window. So we have these 4 plots and we're going to use the par function. We're going to set our plotting window right off the bat. So R makes it pretty easy to combine multiple plots in one overall graph or window. And that's with the par function. So what we're doing here is we're going to set some arguments within the par function. The par function is automatically preset for us every time we open it. So this is what par, it allows you to set graphical parameters. Let's print it out. Okay, so this is what it's looking like right now. It actually has some predefined values, and what we're going to do is we're going to add in our own row options. So right now our par mfro is a 1x1. I don't think it'll show it, but it is a 1x1. Why? Because we only have one plot right here. So if I were to actually go up, if I were to say par mfro equals 1, 2, watch what happens when I plot this. Now we have one row, but we have space for 2 columns. So if I run this again, it'll show up right next to it. So all that I did there, the reason that we're only plotting, we split this into 2 because I specifically said using this function right here, par, I said, okay, hey, graphical parameter named par, I want the mf row function to be defined by 1 row, 2 column. Now, I don't think that these names are particularly good. I don't think that they're very descriptive, but all that you need to know is that mfro right here allows you to specify the number of rows times the number of columns and the plots that are ultimately filled by row. That's why we had first row, or the first column here and the second column here, but if you were to flip it around and have 1 or 2 rows in one column, it would be filled by row. So there's a lot of different options you can have and you can throw in within par. And so one is mar, and that will allow you to specify the margin. You can have the outer margin specified, and these ones actually make a little bit more sense You have the background color, and CEX we kind of talked about earlier. It's the magnification of text and symbols relative to default. So that's what I was saying earlier, that we had a 1.6 all the way back up here. That is times 1.6 times the normal. So that's why we had a little bit of bigger point than we normally have. So anyway, back to our random data set that we made up here. We're going to set all of this. I'm going to copy all of it over, and then I'll go over it over here. So once again, our test 2 looks like this. We have a bunch of spread out values, and we have different levels within them. So now I'm going to copy over all of this crazy code. But really all we want to do is we want to create a 2x2 array within this window. You want 2 rows and 2 columns. Ultimately, we want 4 plots within this window. So we just use mfro at 2, 2. We want the margin to be specified as 3, 2, 2, 1. And if we go back over here, margins is a numerical vector of bottom, left, top, right. So we have a bottom margin of 3, a top margin of 2, left margin of 2, right margin of 1, outer margin of 0, 0, 3, 0, and that is specified the same way, bottom, left, top, right. And ultimately what we're going to do is we're going to add in these plots. So I'll just kind of run through these, and the box function will add in a little box right around that particular plot. So now, because we set our par settings up here, there's our top left, there's our top right, and here we go. Now we finally have an example of it being filled in by row. So it sticks to the row, it fills in that row, and then it goes down and fills in the next row. Add in a box around that one. Add in a legend. Now for the bottom left, we want a box plot. And then we want a pie chart. And then we're just going to add some text, which is going to act as the main title. And we're specifically placing this text in here so that it covers all 4 plots. So we know how plot works. And in this case, what we did is we just set up a basic bar plot and still use the plot function, only we didn't define any y. So what happened is we were just able to plot the infected status, which looks like this. It's just a factor with these levels: infected, recovered, and susceptible. So it counted, because we were in a factor, it was able to count the instances for each level. So we have 12 infected and roughly 23 recovered. And if this was a little bit wider, it would actually show recovered. There we go. And for bar plot, so this is actually a legit bar plot. It's not just using the main plot function. So we have the bar plot function. All of these are just based. They're just automatically brought in. So we wanted to make a table here to be able to show. So again, we did this up above to show the number of genotypes across the infected status. We said we wanted these values to be beside each other, and we just declared these colors right here. So we have green for capital R, or in this case, sea green 4, coral for capital R, lowercase r, and Dodger blue 2 for little r. And again, this box function, all it's doing is adding a bounding box around the specific plot. We add in our legend, and here we go. We have our legend label right here. So I can actually define this. And remember, just as a quick reminder, if you are not going to specify the argument name before using an argument, the function is going to assume that you're using the arguments in their defined order. In other words, if I wasn't defining legend equals right here, I couldn't just throw this first because the function would get confused. However, if I'm actually defining it by name, I can put it in whatever order I want. So we had added in a number of columns there. We said, okay, we only want one column here. We want this to be vertically stacked. And then we added a magnification factor of 0.75. So we wanted it to be to shrink just a little bit. Then we add in our plot right here, our box plot. And what we said is we wanted to just kind of normalize some values against the genotype. And so this is just some arbitrary values that we threw together, just so we actually had a little bit of a spread across these values. And so remember, whenever you see the tilde, it stands for versus is one way to think about it. So we have this normalization, R norm, versus the genotype, and then we color it just the same. And then last but not least, we have this genotype table called capital G, and we just make a pie plot with that using the pie function. And then we just define the colors of each slice of the pie. So yeah, we can go through these pretty quickly, just how the availability of customization for these specific base plotting packages. So pretty straightforward and pretty useful for a lot of different applications. So now we're going to get into tree data, and we're actually going to have to load in a data set, and we're going to perform a little bit of analysis, a little bit of renaming, and then we're going to use the information within this real data set and plot up some of those values. We're also going to use this for the assignment. So let's go ahead and we're going to load this in. Now, one way to load in this data set is if you go to the course GitHub, I have a course datasets folder. Now you can go to that folder and you can download this file individually, but I don't recommend doing this. So you can actually find tree data, click on it, and you can view the raw file. You can right click it, save as a comma separated value file, a CSV. That's one way to do it. But there's an easier way, and you're going to need many of these data sets, and some of these are going to be a real pain to download. So just do this. Come up here, click download code, download as a zip. And now you have all of the code for the entire course, including the data sets. When you unzip that, you will have all the data sets, and just save that folder somewhere. That's all you really need to do here. So that's exactly how I would recommend going about opening and loading all of these data sets. Additionally, R has another way to do it, which is pretty sweet, and that is with the download file function. So I can go over here to R, paste what I pasted over, and we have a URL, which is the GitHub URL to the treedata.csv. So if I copy this, brings you to this nice GitHub display of the data set. I declared a destination file, which is my downloads folder, and I'm going to save it as treedata.csv, and the mode is write. And so I can literally execute this line. It's trying, and boom, it downloaded. So now if I come over here and I go to downloads, treedata.csv downloaded today, which when I'm filming this is January 27th, and boom, there you go. So there's a few different ways to download the particular file that you need. However, I highly recommend when you are at the course GitHub, just downloading all of the code, because when you unzip this file over here, you can go into this, and so boom, look at that. It's exactly the same line up down here, and you already have the data sets. So this is all the data sets right here, and here's treedata.csv. So a few different ways to do it. That's the way I would recommend. Download all the code because this is just going to be very useful when we get into shapefiles. Anyway, back to the tutorial. So we're just going to load that data set up. And we already have a destination that we declared if you use this way. And for me, I have this data set already on my computer, and that is located under Documents, GitHub, Geog 473, 673 datasets. So find your path. Yours will not be users James unless your name is James and you are working on a Mac. And we use the read.csv function to load in this data set. And boom, here's what it looks like. We have tree, we have SPP season, we have is it infected? Is the species infected? We have DBH, sap depth, bark thickness, no bark area, heartwood, and sap area. So now what we're going to do is we are going to manipulate this data frame to make it a little bit easier to work with. And what we're going to do is we're going to say the row names are going to be tree. So let's read in this again. So now, notice that we added in, we got rid of this tree column, which just counts the trees, and we just said that, hey, those are the row names right there. So now we just got rid of that column. So that's how we did that. That's all we did over here. And now we have these different names, SPP, season, infected, DBH, and what we're going to be doing. And then we have these specific, we can actually break it down by variable. So this is the tree.dbh column. So go up here, DBH 42.2, 42.2. So just basic data frame analysis. We're going to rename this variable DBH because it's a little bit cumbersome and not very explanatory. So the fourth column name of tree.dat is DBH, and we're going to rename that to be tree diameter. So we're saying, okay, this is what it is. We're going to rename it as tree diameter. Now we print out tree.dat again. Boom, tree diameter. It's now defined. And now what we want to do is we're going to create a figure that has two plots in a specific window. And we're going to plot a histogram of bark thickness to kind of show the distribution. And then we're going to plot a box plot of sap depth versus SPP. So, which is species. So SPP, species of tree. And so here's what we're going to do. Here's where we're going to actually go ahead and define these. So we're going to define, oops. Oh, I guess I didn't copy it. So now we redefined our graphical parameter settings to be one row, two columns. And now we're going to use the hist function to make a histogram of tree.dat bark thickness. So really quickly, without any customization, this is what it looks like. Okay. But what we really want to do is we're going to add in a label and its bark thickness in centimeters, and we're going to add a title, and then we're going to give it a color. So there we go, histogram of bark thickness. And these are the counts of how many times these particular bark thickness bins occur. So we have 5 instances of bark thickness between 0 and 0.1 centimeters. Then we're just going to add a box plot, which is sap depth versus species. The data set is tree.dat, so we do have to define that whenever we do this. Otherwise, we'd have to do tree.dat $sap depth against tree.dat $SPP. We're going to give it a Y label, a color, name, and boom, there we go. So now we actually have a distribution of sapwood depth by species. So we can see that the Pico species has the largest spread of sapwood depth, and the ABLA species has the lowest sapwood depth by mean and by 75%, 75th percentile. So I go into a little bit more explanation that I just did right here. And now what we're going to do is we're just going to create a bar plot. And so this is kind of an interesting point here. We're going to need to use the table function on tree.sp species, tree.spp. So we have these different levels, and now we can turn it into a table and use the bar plot function right there. And then what we can do, just to go over the count of the different species, we have 5 of each species, is we can customize this bar plot pretty quickly. We can observe the density, blue, the count, Y label is just the count, and the main, and yeah. There's a lot of different customization you can do for base plots. So now all the assignment entails is basically replicating this and this. And the way you're going to do that is you're going to create a plot using the sequence of x values and y, where y is the log of x values. For the line type, use type equals both. So your edges of your PCH is going to be the same color as the line. That's okay. Use a PCH of 21, a CEX of 1.5, and colors of your choosing. You're going to need to set par mfro is 1, 1. You just want one column, one row. Then you're going to make this right here. And first I ask you to rename the SPP variable species and make a 3-column plot. So you want 1 row, 3 column, use your mfro to declare that in your par settings. And you want a sapwood depth histogram, a box plot of bark thickness by species, and a seasonal count bar plot, so the total number of seasons. Use your own colors, borders, line types. It doesn't have to perfectly match this example, but just try to get close to it. Again, use your own colors. Spice it up. Have fun with it. Customize this plot however you want.